How's everybody doing? Fantastic. Excellent. Anybody else doing fantastic? Nobody else is doing fantastic? I'm a little tired. I've had meetings all day long, so it's been a little crazy, but it's good. So, review session tonight in ALS 4001 at 7 o'clock. So I will see you there, or not. You don't have to come, obviously. And I will videotape the thing. So that should all be pretty straightforward. The exam, I posted an old exam online. Let me show you where that is while I'm thinking about it. The old exam is on the schedule page, and you will see exam example here. If you click that, it'll download to your computer. I don't recommend studying old exams, as I've talked about before, all right? But I do recommend especially looking at the exam to understand the format, all right? So there will be 3 sections on the exam. The first section will look to you like a multiple choice, and it's not. So make sure you read through the instructions on that, because you're going to have that section on yours as well. You're going to have that same section on yours. Make sure you read through the instructions. If you're confused or anything, just let me know, whatever. But you'll see it's not a multiple choice. That is, it's not just necessarily one answer. Okay? So read through that section. The other things are pretty straightforward. All right? But I give you that so that you look at the format, and I do recommend you look at the format before you come in. When you come in on Friday, I will ask you to sit in the odd-numbered seats starting 1, 3, 5, 7, 8, or 9, whatever the odd numbers are, moving from that side, okay? If somebody sits in number 2, don't jump over to number 4. Just move to number 3, and I'll ask them in number 2 to move, and so on and so forth, okay? If you have to have a left-handed desk, then there's a few left-handed desks there. If you have to have a right-handed desk, then there's plenty of those around. But I like to spread that out, okay? The way I run the exam is I will give you a full 50 minutes, but I will give you no more. So I'm very picky about stopping at 50 minutes. And the reason I am is to give everybody an equal chance. So when I say stop, then I expect that you will stop. If you continue writing after I say stop, for any reason, you will lose points. So make sure, for example, you've got your name on your exam. A lot of times students say, oh, I'm just writing my name. I said, well, you should have written your name before now. And I do that so that everybody has the same amount of time. It's not so much of a factor in a class like this, but in a class that I teach in the fall that has 400 students, what I discovered was happening was students were sitting in the back so that they could have more time handing in their exams when they got in line, and they were filling answers in as they were coming in, okay? And they were getting, in some cases, as much as 3 or 4 more minutes to work on things that the other students weren't getting. So I said, no, everybody stops at that point. And that's true here as well. So everybody will get 50 minutes. You got to stop when I say stop, okay? And I'll remind you of that on Friday. And I'll give you warning. I'll say you got 2 minutes, et cetera, et cetera. So make sure you got your name on your exam, blah, blah, blah. You'll get tired of hearing me say that. You're probably tired of me already, but that's another matter. So, okay. Questions, comments, concerns, thoughts? Yes? Oh, maybe I've got my old numbers. I had an old edition on there. I didn't realize I had done that. Thanks for letting me know. I will adjust that. But, yeah, make sure that you're following. You'll see in the book where the sections actually run. So thanks for letting me know that. I should have checked that. Okay. The book, as you know, is supplementary. So there's, as I said before, I'm not going to ask you anything that I haven't talked about in class. All right? So there's no, I don't try to trick anybody. I don't do anything with that. I want to know how much you know about stuff. And I want you to have that, hopefully, straightforward for you. Okay? So I'm not going to go find something in the book that I didn't talk about or find something somewhere that I didn't talk about, because that doesn't really test your knowledge of stuff. Okay? Everybody with me? Other questions or comments? Thank you. Okay. Well, we've got some interesting stuff today. We're going to talk a little bit more about the control of enzymes. We might even get done a little bit early. If we get done early, nobody will object, I'm sure. And the control of enzymes, you've already seen some pretty good examples about how it is that enzymes are controlled. So we talked about allosteric control last time, and I'll remind you that allosteric control involves the binding of a small molecule to an enzyme and affecting the enzyme's activity. That can be positive, that can be negative, meaning it can activate or it can inhibit the enzyme. Okay? That's what allosteric control is about. I talked about, or started talking about, another mechanism of controlling enzymatic activity. And that other mechanism was covalent modification. Okay? So when I'm doing covalent modification, I'm changing chemical bonds. The one that I talked about first, last time, or the only one I talked about last time, was putting a phosphate on or taking a phosphate off. Okay? That's a covalent modification. Putting a phosphate on with a kinase, I got the hiccups today. Taking a phosphate off with a phosphatase. Okay? There are other kinds of covalent modifications that can happen to enzymes. But before I talk about the last one, and then we'll talk about how enzymes actually work, before I talk about the last one, I want to say the third mechanism that we have for controlling enzymes. Okay? It's not listed on here, but I will tell you what it is. Okay? The third mechanism is kind of like, well, duh. The third mechanism is whether or not we make the enzyme. Whether or not we make the enzyme. You might say, well, why wouldn't we make the enzyme? Well, not all cells need all enzymes. Our liver, for example, makes glucose. It needs enzymes for making glucose, but my skin cells don't make glucose. They don't need those enzymes. So it's very simple for the skin cells to not be making glucose. They simply don't make the enzymes that make glucose. Okay? So the third mechanism, like I say, it's kind of a duh, but the fact of the matter is it's a very powerful one, is whether or not the enzyme is made. Okay. Well, now I want to go back and talk about a different covalent modification from what I talked about before. So phosphorylation and dephosphorylation, very, very commonly used by many, many enzymes inside of a cell. You saw it with glycogen phosphorylase, and we'll talk about that later in the term. Okay? I want to spend some time talking about a different covalent modification, though, and that different covalent modification is peptide cleavage. All right? So with peptide cleavage, what we're doing is we're using another enzyme to break a peptide bond in a first enzyme. Okay? So peptide cleavage is a control mechanism. It involves a protease. Protease is an enzyme that breaks peptide bonds. Okay? And that protease, if it cuts the right place in an enzyme, can affect that enzyme's activity. Okay? Now, let's talk about this special category. And these are special enzymes called zymogens. I'll explain what a zymogen is in a second. Zymogen, Z-Y-M-O-G-E-N. Okay? What you see is a zymogen that we all have in our body. We all have it. Okay? And we have many zymogens in our body. I'm going to give you some examples, but here's one of them. It's called Chymotrypsinogen. And it sounds an awful lot like an enzyme we've already talked about, which is chymotrypsin. One of the ways that we name zymogens is by putting an O-G-E-N on the end. And something that's a zymogen is an inactive form of an active enzyme. So chymotrypsinogen is the inactive form of the enzyme chymotrypsin. Now, I'll tell you why it's made in an inactive form in a second. But suffice it to say that if I use the phrase chymotrypsinogen, you should know that that's an inactive form of chymotrypsin. Everybody understand that? Okay. Now, why do we have enzymes that are made in an inactive form? There's a very good reason. And chymotrypsinogen is a really good example. And I'll tell you what's going on in this screen picture in just a minute. Okay? First, we need to understand the whys of why we make this, right? We make enzymes in an inactive form because many cases, the place where they're made, they can cause great damage. So chymotrypsinogen is made in your pancreas. In fact, you have many enzymes made in your pancreas that are involved in the process of digestion. If those enzymes that are involved in digestion start doing their thing before they get to the digestive system, you can imagine there would be some problems. And there are. Okay? Chymotrypsin, the active form of the enzyme, is a protease. That is, it will break peptide bonds in other proteins. It's a protease. Okay? If that protease is made in an active form, that is, if it were made in the chymotrypsin form, what would happen as soon as the pancreas cell let it go, the chymotrypsin would turn around and start digesting the proteins on the surface of that pancreatic cell. It would basically kill that pancreatic cell. Okay? So the cell and the body avoids that by making chymotrypsinogen in an inactive form, and then it passes into the digestive system where it gets activated. Does that make sense? Now sometimes, and we know this is the case, sometimes chymotrypsinogen gets activated a little too soon. It gets activated. How does chymotrypsinogen get activated? It gets activated by another protease. So we can imagine that the protease activity might start backing up from the digestive system back in the direction of the pancreas. And that happens sometimes. And when that happens, people develop a condition called pancreatitis. Has anybody ever heard of pancreatitis? Have you ever had pancreatitis? Have you had it? You have? Family did, okay. Pretty painful, from what I understand. Yeah. Pancreatitis can be life-threatening. It's mostly painful, but it can in some cases be life-threatening because what's happening is this sucker is attacking your pancreas. Okay? So there's a really good reason to make this enzyme and other digestive enzymes in an inactive form that gets activated when they make it to the digestive system. Okay? We have other things in our body that are made in an inactive form that cause real problems if they get activated too soon as well. And the most common of these are found in your blood. They're the blood clotting proteins. The blood clotting proteins, they have the same thing. They're made in an inactive form. A protease comes along, cuts a bond, and makes them active. And when it makes them active, they go through a series of reactions that ultimately result in a blood clot. Well, that's great if you cut yourself. You want to have a blood clot there, otherwise you bleed to death, right? It's not so great if that decides to randomly form in your heart or your brain. It's where you develop what's known as a heart attack or a stroke. Okay? So there's severe consequences for the inadvertent or the mistimed activation of Zymogens. Okay? Well, this figure shows how it is that chymotrypsinogen is activated. And it has a lot more information than what we really need. But I'm going to step you through it very briefly and emphasize what I think are the important points. Okay? Chymotrypsinogen is made, as I said, in an inactive form. And there's amino acid number 1, there's amino acid number 245, and no, it's not shown in its folded state. It's only shown as a linear bar just to make it simple to show you. Okay? This enzyme, in reality, is all folded up just like any other globular protein is. But we can see that whole, or we can imagine that whole 245 amino acid piece of chymotrypsinogen looking right in front of us. You'll notice that it's completely inactive, and you might wonder, how is it made completely inactive? And the way it's made is that part of it covers up the active site. The way that it's made in one intact piece, the active site is buried in the enzyme. There's no way the substrate can get to it. The enzyme gets activated by an enzyme called trypsin. Trypsin is a protease. And yes, all this is important so far. Trypsin is a protease. It cleaves peptide bonds, and it cleaves one peptide bond. It's the peptide bond between amino acid 15 and 16, and no, I don't care if you know it's between 15 and 16. But it cleaves one peptide bond. That peptide bond now allows an opening into the active site. It's a very tiny opening, but it allows an opening into the active site that didn't exist before that peptide bond got cleaved. Well, that little tiny opening allows this enzyme to be partly active. And the partly active enzyme we call pi chymotrypsin. This pi symbol followed by chymotrypsin. Pi chymotrypsin will cleave similar pi chymotrypsins and cut out some pieces. And you can see some of those pieces now have been completely cut out of the enzyme. And the enzyme is held together by, what do you suppose is holding these pieces together? Not peptide bonds. What do you suppose is left holding those pieces together? It's another covalent bond that we find inside of proteins. Disulfide bonds. These are disulfide that's holding everything together. Well, the beauty of this is the active site is wide open and ready for business. This enzyme at this point is fully functional and capable of attacking any protein and making cuts in the peptide bonds, breaking peptide bonds. Well, this is pretty important. So this process is happening in your digestive system, and in your digestive system, it takes off and does its thing. You might say, well, doesn't the digestive system get attacked by these enzymes as well? What do you think? Answer is yes, it does. Some of the most rapidly replicating cells in your body are in your digestive system because they do get damaged by things. People get ulcers and people have problems with their digestive system because something is interfering with their ability to replicate and make more digestive cells. If that happens, you get ulcers, you get problems associated with your digestive system. But the digestive system is set up to take care of that by making more cells quickly. The pancreas is not. Everybody with me? Yeah. Yep. It does. It digests itself, yeah. Yep. Pi chymotrypsin is a protease, that's correct, as is chymotrypsin itself. Pretty cool. Other questions? Okay. Well, that's what Zymogens are all about. We don't have time in this class to go through the blood clotting scheme, but the blood clotting scheme is really remarkable. There's about 15 different proteases. One activates the next one, activates the next one, activates the next one, activates the next one. And you get a whole cascade of these that happen. There's a whole chain reaction to this. And the net effect of that is that you make a lot of the final one. And the final one will form polymers. And it's those polymers that give rise to clots. So the reason we have in the blood clotting system, we have this whole chain of events, is that every step we get more. So we get an amplification, and then we get an amplification of the amplification, and an amplification of the amplification of the amplification. And so that it gets bigger and bigger and bigger. so that this process happens very rapidly, and that's how a clot can form before you bleed to death. The speed with which that happens is important, and you have to make a lot of these things. A clot is really a remarkable thing. It forms without your being aware of it. It starts forming in seconds after you cut yourself. It forms a completely watertight barrier that blood or other things can't go through. That's pretty remarkable. And it's flowing inside you right now. You can imagine if we have something that causes that signal to get screwed up, that's how we're going to get heart attack, stroke, etc., because amplification of amplification of amplification can cause a quick problem somewhere else, right? Okay. Well, that's not what I'm here to talk about, so I should shut up about that. What I want to do now is talk a little bit about how chymotrypsin works. And chymotrypsin is going to be an example or a model enzyme for a group of enzymes that we call serine proteases. Serine proteases. And I'm going to explain that to you in just a minute. Okay? Let's think about studying how chymotrypsin does what it does. Okay? Now, I like to say, and I'm a biochemist, so I'm allowed to say this, that biochemists are really lazy people. Okay? Biochemists are really lazy people. We want to do our experiments in ways that don't involve too much work. We're not unlike anybody else, right? We like to do our work in the easiest way that we can. And in the case of chymotrypsin, what we're trying to study is a protease. And measuring the rate with which a peptide bond gets broken, if we don't do any special tricks, is kind of a tricky thing. It's very time-consuming, and biochemists being lazy like to get their answers and results quickly. So what biochemists have done is they've found artificial substrates that look to chymotrypsin like they're a peptide bond. In other words, we can fool chymotrypsin into thinking that this thing that we're giving it is a peptide bond that it can break. Now, the lazy part's coming up. This material that we give chymotrypsin starts out colorless. Chymotrypsin attacks it, breaks the bond in it, and when it breaks the bond in this thing that we've given it, it turns yellow. Well, that's really easy for us to study. We can put this in an instrument called a spectrophotometer and determine how much yellow is produced, how fast. Okay? Laziness really helps us out here. Okay? Well, when we do this with chymotrypsin, we discover something kind of surprising. The surprising thing that we discover is that the reaction starts off, this is velocity moving on the y-axis, this is time over here. Okay? We discover that the velocity goes up really quickly, and then it kind of levels off. And that's not the same thing as we saw in the V versus S, because in the V versus S, we were saturating the enzyme with substrate. We were plotting substrate. Here we're plotting time. Why does that change over time? Why doesn't it just go linear? Why does it have two components, a fast and a slow component? Well, the answer to that question is that the mechanism of the reaction occurs in two steps, a fast step and a slow step. A fast step and a slow step. I'm going to explain to you and show you what that fast step and slow step is in just a minute. Everybody understand that there's two steps to this reaction? Okay. Here's the enzyme, shown by the E. Here is this artificial substrate that we've given it that is given by this big name that you don't need to know. See, if I don't repeat it, then I haven't actually technically said it, and if I don't say it, then you're not required to know it, right? So if you keep me from saying these things, then you'll never have to know anything, right? Okay. All right. What we see are two steps that happen. A first step that happens very rapidly, and a second step that happens very slowly. We're going to see the actual mechanism it goes through in just a second, but this is a simplified version of it. The first step, the enzyme recognizes the substrate. And this is true whether it's a protein or it's this artificial substrate. It doesn't matter. It's the same for both of them. The enzyme binds to this guy, and it cleaves a bond. You see the bond right here? This bond right here where my pointer is gets broken, and this guy right here on the right, whose name I'm not mentioning, gets released. This is yellow. This is colorless. So very quickly, the first thing that happens is yellow is released. And that's that boing we saw in the plot, the velocity, the very quick initial velocity. But something has happened here. The enzyme didn't just break a bond. We discover that what happened was the enzyme broke the bond, but in breaking the bond, it became attached to a part of that molecule. The slow step of the reaction involves the release of the enzyme from that molecule. Fast step, breaking the bond. Second step, getting released from that molecule. Okay? I always come back to relationships, but you've probably had relationships where you very quickly got into something that seemed great at the time, but getting out of that sucker may have been a real pain, right? Relationships have two phases, just like some enzymes have two phases. This enzyme has two phases. The getting out of the relationship part of it is right here. Okay? You'll see water is there, and as we're going to see in the mechanism, water is needed to release the enzyme from this part of the molecule. Okay? Everybody with me? All right. Here's what happens. Oh, boy. Now, I'm going to step you through this. It's going to look very techy, and you're going to say, oh, my God, am I going to memorize all these steps? And the answer is, I am not going to ask you to draw this mechanism on an exam. Okay? I am not going to ask you to draw this mechanism on an exam. I'm going to tell you what this mechanism is, but I'm already telling you here, and it's on videotape, I'm not going to ask you to draw this mechanism on an exam. Okay? Everybody with me? So don't get overwhelmed by what I'm going to tell you, because what I'm going to do is I'm going to step you through it, and then I'm going to back away and say, here's the bottom line. Everybody with me? First thing is something that you need to know. The first thing is that the active site of the enzyme has 3 important amino acids in it. 3 important amino acids in it. Okay? Only 2 of them are shown on this figure. Okay? One's way over here on the right. The 3 things on this, the 3 amino acids are called the catalytic triad. The catalytic triad. These 3 amino acids are the side chain of serine, the side chain of histidine, and the side chain of aspartic acid, or aspartate. Same thing. The side chains of serine, histidine, and aspartic acid. Chemical reactions, you all should know, of course, involve changes, swapping, movement, bonding of electrons. Enzymes, when they catalyze reactions, favor movements, bonding, linkage of electrons. Okay? So what we're going to see in this mechanism I'm going to tell you is going to involve some shuffling, shifting of electrons to make some reactions occur. Okay? Here's what happens. First, The active site is where the substrate binds, right? I told you before, you may not remember, but I'll remind you here, that chymotrypsin prefers to cut next to certain amino acids. I didn't tell you what they were. I didn't require you to memorize them, at least for chymotrypsin. But there are several that it prefers. So let's imagine that chymotrypsin is sitting here, and along comes this polypeptide, this protein. And the proper amino acid pops into chymotrypsin and is sitting in the active site. What's going to happen? Well, I hope what you would tell me would happen would be the following thing. The binding of the proper substrate causes the enzyme to change shape slightly. Right? There's your induced fit. The binding of the proper substrate causes the enzyme to change shape slightly. The change in shape causes some very, very minor changes in the position of the 3 amino acids I just told you. Yeah, mechanisms are a pain. I'm not a fan of mechanisms either. But this is kind of cool what this does. Changing the shape. The 3 amino acids. Aspartic acid sitting over here, not shown. Histidine right here. And serine right there. They each play a critical role. Aspartic acid is a carboxyl side chain. What happens to carboxyl side chains at physiological pH? I'll tell you. They ionize and make a negative charge. The aspartate that's in this active site has a side chain with a negative charge. That negative charge, when we have this slight shape change, that negative charge of aspartic acid has moved very close to this ring of histidine. Do electrons like negatives or do they dislike negatives? They dislike because they're negatively charged themselves, right? The electrons in this ring decide, I'm getting away from this negative charge. They move to the left side of this ring. And when they move to the left side of this ring, they are partially negatively charged. And what do negatively charged things happen to like an awful lot? Positively charged things. And what's the closest positively charged thing? It's that proton on the side chain of serine. And so the histidine grabs that proton away from serine. That leaves behind an oxygen that has lost a hydrogen and now is negatively charged. A negatively charged oxygen made in this way is called an alkoxide ion. That's a name you need to know. Alkoxide. Alkoxide. An alkoxide ion is made by pulling a proton off of a hydroxyl. You've seen now how that proton got pulled off. Okay? This is all happening very rapidly. Very rapidly, instantaneously. Okay? That hydroxyl that is negatively charged is extremely reactive. It will make a chemical bond with the first thing that it bumps into. And the first thing that it bumps into happens to be the polypeptide that bound in the active site. That alkoxide ion attacks the peptide bond. Okay? That alkoxide ion is something you referred to in organic chemistry as a nucleophile. It's looking for a nucleus, and the nucleus that it finds is the carbon of the peptide bond. When it attacks the carbon lets go of the nitrogen it's attached to. You have just broken the peptide bond. This reaction happens rapidly. This is the fast part of the reaction. You've just seen the fast part go boing. Binding, shifting, loss of proton, attack of the peptide bond. Bang, 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 that happened very rapidly. The first part of the reaction is done. Part of the protein goes flying away. You can see it flying away here because it's not attached to anything. Everybody with me? Not attached to anything, okay? Well, the serine, on the other hand, is attached to something. It's attached to the remainder of that peptide chain. We've let half of it go away, but the other half is attached to serine. The rest of this involves water coming in and releasing the peptide from serine, and that's a slow step. Fast step, slow step. Okay, so that's the mechanism about what happens. Now I'm going to give you, as I said, the bottom line. Here's the bottom line of what happens. First, binding of the proper substrate. Second, shape change in the enzyme. Third, creation of the alkoxide ion. Fourth, attack of the peptide bond by the alkoxide ion. Fifth, release of one of the peptides. One of the peptides, the piece that got broken off. Sixth, the release of the first peptide from the serine. It's that last step, that's the slow one. That requires water. That's what's happening here. Now, I show this to you as an example, not to confuse you, not to over-dazzle you with any of this stuff, but to show you something that an enzyme is doing in catalyzing a reaction. What an enzyme is doing in catalyzing a reaction, this is being made possible by that slight shape change that we described before. I described it the last time I talked about it as stress or tension. In this case, the stress or tension involves electronic stress or tension because it's the electronic stress or tension that allows that proton to be pulled off. And it gets pulled off because everything moves closer together on the binding of the proper substrate. The flexibility of enzymes is what makes them magical. The flexibility. Because enzymes are flexible, all these things happen. Questions on what I've told you? Nobody? Yeah. An example of where this occurs. So this happens every single time that chymotrypsin breaks a peptide bond. This series of cyclic steps. And at the end of this, when we end up at the end, we have the serine right back where it was with an OH on there. We return the enzyme to its original condition. And if you remember your definition of a catalyst in your chemistry class, what they said was a catalyst a catalyst speeds a reaction but is not changed in the process. Right? The enzyme, in the end, is not changed. But we see that the enzyme can be transiently changed. That's part of that induced fit mechanism that we talked about. But it gets back to its original state. It's pretty cool. I'll tell you something you don't need to know. How about that? Okay? This way you can relax. So the song I did on enzymes the other day talked about suicide inhibition. Did anybody notice that in the song? Okay? So you don't need to know this, but I'll tell you. So suicide inhibition is actually a way to kill an enzyme. We've talked about inhibiting an enzyme. And there's many examples of drugs that do that. There are also drugs that kill enzymes. And they kill enzymes by virtue of the fact that the enzyme binds them. The enzyme starts to catalyze the reaction, but because it's not the proper substrate, this inhibitor gets covalently attached to the active site. The enzyme commits suicide because by trying to do its thing, it gets stuck with something covalently bound to its active site that it can't get rid of. Okay? That mechanism is how penicillin works. Penicillin works by covalently binding to an enzyme, and no, you don't need to know this, I'm just telling you this. It works by covalently binding to an enzyme that the bacteria need to make cell walls. When you give a penicillin to a bacterium, it gets covalently, its enzyme gets covalently bound to penicillin and can't make cell walls. If it can't make cell walls, you can't divide. Pretty cool. Okay? Now we'll go back to the things that you're responsible for. How's that? Okay. There we are. That's what it all looks like. There's, it's sort of twisted around from what I had before, but there's serine, there's histidine, there's aspartate. If you rotated that about 90 degrees, you'd see it in the orientation that I described to you. Okay. Here's another protease. Oh my God, he's going to give us more, right? Well, I've already shown you one. This one's going to be really, really simple. It's going to take about 2 minutes to explain to you. The last one I described to you is what I called a serine protease. And there are a bunch of serine proteases, and it's also coming back to your question about where do we see this. Many of the proteases that we have in our digestive system are serine proteases. They use exactly the same mechanism as chymotrypsin uses. Trypsin, for example, is a serine protease. There are many other serine proteases that we have in our cells. Okay? Besides serine proteases, there are closely related proteases called cysteine proteases. So the serine protease, you saw that the side chain of serine played a very important role in the catalysis. It formed that alkoxide ion, right? Cysteine proteases have an activated sulfur that happens, and the activation happens in exactly the same way. Histidine comes along, pulls a proton off of an SH group. The SH is the sulfur that's left behind is very reactive. It attacks the peptide bond in exactly the same way. And the same series of steps that we saw in the serine proteases happens. The only difference being that we have an SH instead of an OH. Now you've got two proteases down. Okay. Let's see. What else can I say here? I said I would get you out early, didn't I? We'll talk about coenzymes next time. I do have a song for you. It's about serine proteases. This one I can actually sing a little bit, and you can sing with me. It's a very easy one to sing. It's called the New Serine Protease Song. If you, I have a deal for you. If you will sing very loudly, there will be an extra credit question on the exam. Is that a deal? Okay. All serine proteases work almost identically using amino acid triads catalytically. First they bind peptide substrates, holding on to them so tight, changing their structure when they get them in the S1 site. Then there are electron shifts at the active site. Serine gives up its proton as the reaction goes on. Next, the alkoxide ion, being so electron-rich, grabs peptide's carbonyl group, breaks its bond without a hitch. So one piece is bound to it, the other gets set free. Water has to act next to let the final fragment loose. Then it's back where it started, waiting for a peptide chain that it can bind itself to. Go and start all over again. Okay, guys. Review session tonight. Good luck on the exam.